Okay, so in this chapter, we've explored how cells interact with each other via these social networks. And specifically, we looked at these connections and communication between adjacent cells. Next, what we're going to be looking at is how distant cells communicate. And we're going to do this by taking a close look at signaling in multicellular organisms. So distant cells communicate, as I just said, by using signaling molecules. And sometimes signaling molecules are generically called ligands. So sometimes I might be referring to a signaling molecule and use the word ligand. Different groups of signaling molecules include neurotransmitters. These are signaling molecules that regulate the nervous system. And hormones. Okay, hormones are signaling molecules that are sent to distant organs and regulate physiology and behavior. Signaling molecules can also include growth factors. These provide signals for cells to divide, and cytokines, which are signaling molecules that regulate the immune response. And so today we're really going to consider the signaling molecule that are hormones, okay? And so hormones are categorized based on their ability to cross the plasma membrane. And so we can categorize lipid hormones as lipid soluble, like a steroid hormone like testosterone, or insoluble like epinephrine. And so we're going to take a look at these two different signaling pathways. But already you're probably starting to think that those that are lipid soluble might be able to pass through the membrane. And so their receptor is probably inside the cell. And those that are insoluble have to probably rely on some sort of protein or receptor on the outside of the cell to begin sending their signal. So let's first take a look at this lipid soluble signaling molecule pathway. And so here let's consider that this is testosterone, and this is carried through the blood, and it's attached to a protein, right? Because it's not very hydrophilic. But once it gets to the surface of the cell, that hormone can diffuse right through the plasma membrane. So stage 1 is the arrival of the signal, and stage 2 is signal entry, right? So this goes straight through the membrane, and it binds to a receptor in the cytosol. And when it does that, it causes the receptor to change shape. And so this is a really common theme in signaling, and we call this signal reception. And so you can think of this as here is our quarterback, and they've thrown this signal, right, straight through to a receiver. And then that receiver, right, is going to go right into the end zone. And in this case, our end zone, right, is the nucleus. So this shape change may have exposed a nuclear localization signal. So it might have been hidden in the interior of this protein. And then when it bound to the ligand or bound to the hormone, this caused a shape change. So now important can see the nuclear localization signal, and then this can be brought into the nucleus, where it can regulate gene expression. So we have the arrival of the signal and signal entry, and then we have signal reception and signal response. Okay, so very few steps in this signaling pathway when something can pass through the receptor or pass through the plasma membrane. So very short, easy play that ends in a touchdown. Signaling gets a little more complicated when we start to think about these lipid insoluble molecules. Okay, so I want to talk about the steps here. So the first step is that our extracellular signaling molecule binds to a receptor protein in the plasma membrane. Okay, so this is our first signal reception. Okay, so here, right, we're passing this ball, right, to a player, right, maybe a running back. Okay, and I'm sorry for all the football analogies. Okay, this binding causes a shape change in this receptor. And now this is going to trigger a cascade of events that we refer to as signal transduction. Okay, so this is an important term. So we have signal reception, and then we have signal transduction, because what we're doing here is we are changing the language by which this signaling molecule is communicating, right? So we're going to transduce this signal. So it's going to convert the extracellular hormone signal into an intracellular signal. And when it converts this, it will often amplify this signal. Okay, so one player, right, now becomes a little bit of a Ponzi scheme where one person can enlist a few more people, and then those people can enlist a few more people. And so you amplify this signal, okay? And this lets the body get away with producing very low levels of hormones to produce a very strong intracellular response. This pathway, just like the other pathway, eventually leads to a signal response. And often this can be changes in gene expression, but it can also be a signal response that causes movement or the mobilization of stored vesicles. But we're mostly going to think about how signal reception leads to changes in gene expression.
So the example we're going to use to talk about lipid-insoluble signals is epinephrine. So when there's a loud noise that scares you, your neurons will detect this, and they'll send a signal to your adrenal glands. And your adrenal glands will start producing the hormone, the lipid-insoluble hormone, epinephrine. And epinephrine will be picked up as a signal by multiple cells throughout your body. And they will coordinate this response, this whole organism response, to make more ATP. And that ATP will provide the energy to sustain this flight or fight response. Now, before I get into the details of this pathway, I want to introduce you to some key terms. So the first molecule I want to talk about is GTP, okay? GTP is very similar to ATP. It's got it's a nucleotide. It's got a ribose sugar and 3 phosphates. But instead of having adenine, right, which would make it ATP, adenine triphosphate, this has a guanine. So we're substituting the base when we're talking about GTP. So this is guanine, a ribose sugar, and 3 phosphates, and it's a nucleotide. Now, the other person that player I want to introduce you to is a G protein. So these are common proteins that work in signaling pathways, and they refer to any type of protein that is activated when it's bound to GTP. Okay, so a G protein just means it's a protein that's activated when it binds to GTP. These G proteins are turned off when GTP is hydrolyzed, okay? So what does that mean, the hydrolysis of GTP? That is when a phosphate is removed from GTP, so it has 3 phosphates, and when it's removed, it becomes GDP. So that would be guanine, a ribose sugar, and 2 phosphates, and it's often shown as GDP plus an inorganic phosphate. So I'm going to be talking about the hydrolysis of GTP, and that's all I'm talking about is the removal of one of those phosphates, and then when that happens, it becomes GDP. In other signaling pathways, we're going to learn about these enzymes called kinases and phosphatases, okay? So kinases are enzymes that add a phosphate, usually from ATP, to a substrate. So essentially substrate level phosphorylation. And a phosphatase is an enzyme that then removes that phosphate from the substrate. So kinase and phosphatases have opposite roles in the cell. One's adding a phosphate and the other one's removing it. Okay, so I just wanted to introduce you to these terms because they're going to come up a couple times in the next examples.
Let's take a look at this with a real pathway, okay? And the pathway that we're going to really focus on is this pathway called G protein coupled receptor pathway, okay? So this is a G protein coupled receptor. And when these were originally identified, they were called serpentine receptors. because each one of these in our body has 7 transmembrane domains. And so this protein snakes through the plasma membrane several times, right? Snakes through it 7 times because you have the seven transmembrane portions, and then you have these intercellular motifs and these extracellular domains, okay? And the ligand is going to bind in the extracellular space, and it's going to transduce the signal into the intercellular space. We also call these G protein coupled receptors, we call them allosteric proteins, because when their ligand binds, they're going to change shape. So let's show that here. I'm showing the ligand as this green circle, and we can think of this as epinephrine binding to the beta-adrenergic G protein coupled receptor. And you can see, right, that this protein has changed shape, okay? And sometimes we depict this as a receptor with a little star to show that it's been activated. Now, this is going to transduce a signal, okay? And the signal it transduces, right, is part of its name. So G protein coupled receptor. So it's actually coupled to a G protein. And the G protein that it's coupled to is actually a trimeric G protein. So there's 3 parts of this protein. There's this alpha, beta, and gamma subunit. So that's why it's called trimeric, okay? So just to review, we have our G protein coupled receptor. When the ligand binds, it changes shape, and then it's coupled to a trimeric G protein. Okay, so recall that G proteins are only active when they're bound to GTP, okay? And they're inactive when they're bound to GDP. So initially, before the ligand bound, these were all associated, and the alpha subunit is bound to GDP, okay? But what this receptor is going to do is it's going to promote the exchange of GDP for GTP. And so this alpha subunit is going to bind to GTP. So now the alpha subunit has GTP, and this is going to change the shape of the alpha subunit, and it's going to cause it to dissociate from the beta gamma subunit. Okay, so let's do this one more time. We have our G protein coupled receptor, the ligand binds, it changes shape, right? This shape change helps the alpha subunit of the trimeric G protein exchange GDP for GTP, and this causes the alpha subunit to dissociate from the beta gamma. Okay. Now next is where I think it kind of gets a little more interesting, because this alpha subunit is going to go ahead and activate an enzyme. And the enzyme it activates, and you don't have to know the name of this enzyme, but it's called adenyl cyclase. And so this alpha subunit activates this enzyme. Now, this is the important part, is that this enzyme is going to lead to the production of a secondary messenger. Okay, so let's just take a second to define what a secondary messenger is. It's a small non-protein molecule that relays and amplifies signals received from the surface receptor. Okay, and the surface receptor is the primary messenger. Okay, so there's several features of it. It's non-protein, and it's usually able to amplify the signal. Okay, and so in this case, with this example, adenyl cyclase is going to convert ATP to cyclic AMP. And so here's what the cyclic AMP looks like, right? So the ATP has lost all of its phosphates, and now, all but one, and then it's connected, right, in this kind of cyclical fashion to the sugar. And this cyclic AMP is a secondary messenger, okay? And this is going to go ahead and activate other proteins, okay? I'm going to give one example of what cyclic AMP activates, but they aren't restricted to one role, and several may act in a single pathway. So I'm only focusing on cyclic AMP, but several could be acting downstream of G protein coupled receptors. And so examples of these small molecules right, include things like cyclic AMP, as well as calcium ions that are stored in the endoplasmic reticulum. This should say DAG. So DAG and IP3 are lipid signaling molecules. So they're non-protein small molecules that amplify signals. Okay, so these are lipids, this is an ion, and this is a metabolite of ATP. So what does cyclic AMP do? Well, it activates, so it binds to this regulatory subunit on a particular kinase, and I'm not going to give you the name of the kinase, but it then activates that kinase. So what kinases do is they add phosphates to other substrates. So once this is active, this kinase is active, what it does is that it's going to be able now to move into the nucleus. So a nuclear localization signal was exposed. And it's going to go into the nucleus and it's going to phosphorylate transcription factors. And then those transcription factors, once they're phosphorylated, can turn on different gene expression. And so we have ligand, a change in shape, that facilitates the exchange of GDP for GTP, right? So that the trimeric G proteins break apart. This activates an enzyme that leads to the production of secondary messengers, including cyclic AMP. And cyclic AMP activates a kinase that leads to gene expression. A common theme I'll point out is in signaling pathways, we see a lot of kinase activation. So adding phosphate groups to proteins is a really easy way to turn proteins on and off very quickly. And so it's quite common to see kinases in pathways, and we'll take a look really quickly at another pathway that really only uses kinases. So if we start with a single molecule of epinephrine, right, this can bind to a G protein, right? So we have one GPCR, right, binding to epinephrine, can actually activate hundreds of G proteins. Okay, so there's an amplification event there. But each G protein only activates one adenyl cyclase. Okay, so here we have a lot of G proteins, but each G protein activates only one adenyl cyclase. But then each adenyl cyclase makes many cyclic AMPs. So cyclic AMP its production represents a signal amplification, right? We're turning up the volume on this stereo, right? So that we can really produce a strong response. Just to give you some numbers, the blood levels, just kind of for 1-to-1, of epinephrine are 10 to the negative 10th molar, so minute amounts. And the cyclic AMP levels inside the cell, once that cell has bound to epinephrine, is 10 to the negative 6. So that's 4 times higher, right? So we've really cranked up the volume. And so this helps make this process far more sensitive. So I said we triggered a response, a gene response. And so what would that response look like if we were considering the effects of epinephrine? Okay, so these bind to a group of receptors called adrenergic receptors. And there's different adrenergic receptors on different cells. But in theory, in overall, they're all going to trigger some metabolic changes, okay? So in liver and muscle cells, binding of epinephrine to their adrenergic receptors is going to stimulate glycogen breakdown. So it's going to cause glucose to be released. And it's also going to increase, the body's going to increase blood glucose levels. And the more glucose means the more ATP. In muscle cells, those cells will also start upregulating the enzymes in glycolysis. So those are some of the genes turned on. And so, again, this all helps make more ATP, more energy, so that you can run away. If epinephrine binds to heart cells, this increases the heart rate by vasodilations in the blood vessels. And it also increases, if it binds to cells in the lung, it increases the respiratory rate via bronchodilation. So this means more oxygen so that this oxygen can be used by the electron transport chain to make more ATP. Now, I don't expect you guys to definitely memorize this list, but I just want to give you a connection to how a signal, right, can transmit into something that's useful for a multicellular organism. So every signaling pathway needs to be regulated. We don't need to break down all of our glycogen, right? Some of it needs to be stored, right? We only need that short burst. And so I just want to talk about some ways that this pathway can be turned off. So every time an alpha subunit turns on an adenyl cyclase, its GTP will be hydrolyzed to GDP. And so what will happen is then when it's hydrolyzed to GDP, it will actually reform as a trimeric G protein. Okay, it can be reactivated if that hormone is still bound, but it can only activate one adenyl cyclase. Okay, so that's short-lived, seconds to minutes. So that's one way the pathway can be regulated, right, is this gets turned off. The other way is that cyclic AMP can also be converted into an inactive product. Okay, so this is AMP, and so you can see that the cyclic portion has gone away. And this happens via an enzyme. Also, right, the hormone or ligand can dissociate from the receptor, and this would change its shape back so that it wouldn't be able to associate with the trimeric G proteins. So these are some ways that this pathway can be naturally turned off. And then the last way I'll talk about is that there's a lot of proteins in the cells that are called phosphatases. And phosphatases have the opposite activity of a kinase. So these are kind of two opposing proteins because this is an enzyme that removes phosphorylation. And so phosphatases can take off the phosphate from the transcription factor that's causing the responses. So this pathway can be naturally turned off. The whole signaling pathway is quite dynamic. So in a cell that's signaling, the numbers of receptors can change, the ability of the receptor to bind to a hormone can change, and the sensitivity to a hormone can change. So this is a very dynamic process that's very well controlled. Signaling can also be actively blocked, and this is why I chose, or one of the reasons we talk about G protein coupled receptors is because 1/3 of all FDA approved drugs target a G protein coupled receptor. So it's a pretty pharmaceutically important pathway to know about. And so, for example, we can add a competitive inhibitor. So in the case of epinephrine, we can add a beta blocker. So recall that competitive inhibitors bind within the active site of the protein. So they bind where the hormone would normally bind. And if you bound here, this wouldn't transduce this shape change. And so you wouldn't get activation of your trimeric G proteins. You wouldn't get activation of your cyclic AMP. So you wouldn't get, we call this downstream in signaling. You wouldn't get any of these downstream pathways happening. And this drug is used to reduce the strength of heart contractions to lower blood pressure. There's different types of inhibitors for G protein coupled receptors. We can get allosteric inhibitors that bind somewhere else in the protein and shut it off. And we can also inhibit downstream signals. So for example, if we inhibited adenyl cyclase, right, it wouldn't matter if there was hormone around. If we inhibited adenyl cyclase, we wouldn't get any cyclic AMP. So that's what I mean by inhibiting downstream signals.
So in summary, we have the ligand binding to the G protein coupled receptor, and that's the signal reception. When this happens, the G protein coupled receptor undergoes a conformational change that initiates the next stage, which is signal transduction. So that GPCR with the star, meaning that it's changed its shape, it activates G alpha. So that's a G protein, which means it's bound to GTP when it's active. And then that allows it to dissociate from beta gamma. And then this leads to the G alpha activation, leads to eventually the production of a secondary messenger called cyclic AMP. And cyclic AMP amplifies the signal. So we have a lot of signal amplification going on. And then eventually that will activate kinases, which will then exert a variety of signal responses. And so in the case of epinephrine, epinephrine will cause some cells to release glucose from glycogen stores, other cells to increase glucose uptake by changing the levels of their transporters. A lot of cells will increase glucose metabolism by upregulating glycolysis enzymes. And then other cells, when they see epinephrine, will speed up blood flow so that more oxygen can be taken into the body, and that oxygen can then be available for aerobic cellular respiration. And so this is kind of a big picture of signaling response from the beginning to the end of how this happens with G protein coupled receptors. There's a lot of other signaling pathways that we won't have time to talk about. Another common one that's mentioned in the textbook is this enzyme-linked receptor pathway. There's still signal reception, there's signaling transduction that occurs, and then the signal response. In this pathway, this is called an enzyme-linked receptor pathway. The most common enzyme-linked receptor pathways are called these receptor tyrosine kinases, and they bind growth factors. They play a very important role in cancer because if cells sense these growth factors when they shouldn't, then this can lead to cells dividing when it's not the right time to divide. But the way the pathway normally works is that these signaling molecules bind to a receptor. That receptor then dimerizes. This means that two subunits come together and connect. And when this happens, this receptor, it's a kinase, so it becomes autophosphorylated. So you'll see here's that ATP adding phosphates to both of the receptor chains. And then that phosphate then can activate a G protein. And then that G protein then activates more kinases. And what we get is a kinase cascade where the activation of one kinase activates another, and so on until you trigger a response. And so we might in class compare a little bit between the receptor tyrosine kinase pathway and the G protein coupled receptor pathway. The G protein coupled receptor pathway we're going to cover in more depth, but if I ask you about this enzyme linked receptor pathway on an exam, you would see this diagram. The reason I'm bringing this up is because more than 30% of all human cancers are driven by mutations in this receptor tyrosine kinase pathway, particularly in this G protein that's an intermediate in the pathway called RAS. And it's particularly problematic in pancreatic cancer, where 95% of people have a mutation in KRAS that causes this pathway to be essentially chronically active. So even in the absence of this receptor dimerization or in the absence of growth factors, this KRAS is on, it's bound to GTP, and it's causing this phosphorylation cascade, which culminates in a response for the cells to divide. And that's really problematic, and this could be a potential cancer therapeutic is targeting KRAS. Okay, so the last thing I'll talk about is this idea of signal crosstalk. And that it's really important to realize that in multicellular organisms, a single cell isn't receiving a cue from one ligand alone. So, for example, a cell may be sensing epinephrine at the same time as it's sensing testosterone or estrogen or a bunch of signals, and it has to kind of connect these and integrate these signals to respond appropriately. And what that means is that these signaling pathways are usually connected into a network. And so let's take a look at this here in this diagram. So we have signaling molecule A that binds to receptor A, and receptor A then triggers a signal transduction pathway that leads to cell response A. Okay, so let's imagine that a cell sees both A and signaling molecule A and signaling molecule B at the same time. Now, alone signal molecule B, it goes all the way from signal reception to cell response B. But in the presence of A, one of these intermediates actually inhibits receptor B. And so if A is present, it can actually shut down any of the signaling that occurs from receptor B. Okay, but let's make this a little bit more complicated and add signaling molecule C. Now, when A, B, and C are present, if you follow this pathway, you can see that C actually stimulates B2, okay? Now, B2 is downstream from the receptor. And so even if A were to shut off the receptor, it doesn't really matter because C will stimulate a downstream molecule in this pathway. And so this pathway will run if C is present. Okay, so A can no longer shut it down. And we'll get a little bit of practice interpreting these diagrams in class. So in summary, cell communication is a fundamental feature of cells. Lipid-soluble signaling molecules are received by intracellular receptors, and lipid-insoluble signaling molecules are received by receptors on the cell surface, and then that signal is transduced inside the cell. This usually involves some sort of conformational change in the receptor, as well as either the production of secondary messengers and or the activations of kinases. And like with all processes in biology, it's important that we regulate these processes and we make sure that they're turned on when it's only beneficial for the organism to be turned on. And so signaling is regulated. We also have to be able to integrate these signals. And we can, if we understand how signaling works, we can block these signals in therapies in different diseases. So that's it for this video, and I look forward to seeing everyone in class. Thanks, bye.